Hello class. Today we are going to start our first chapter of BIO461 molecular biology course. So let's have a look at a quick look at the syllabus. So the chapter 1 of the syllabus is focused on history and overview, and it has various subtopics. DNA is the genetic material, the discovery of DNA double helix, the DNA structure and function, DNA replication, semi-conservative mode, and DNA double helix can be reversibly melted. Flow of genetic information, transcription, translation, and genetic code. So as you note that this chapter talks about history. Now, history is a very important part of science because there is always a starting point where a scientist observes some facts, and those facts becomes a point of further question. And depending upon the tools and technology at that time, scientists try to probe and get the answers. The new answers give rise to further new questions. So it's a process by which science has developed. Now, if you note that most of the discovery of on biological sciences started to happen at Europe, because Europe was the center of education and science and research at that time. So, and slowly these invention, discoveries, questions, they all came to US, and US became the leader for scientific discovery, particularly in biomedical sciences. So let's start this chapter with a brief look of history. And history is very important so that you know what is the starting point, okay? So before I go into this chapter, so this is the book that is a very good book, very important book for this course. And this is what has been mentioned, 4th edition, Essential Biology by Alberts. So as I mentioned to you, we go to the history. So in as early as 1595, Zacharias Jensen created the first compound microscope. In 1655, Robert Hooke, using that microscope, he devised and looked at the cell walls of the cork for the first time. And Robert Hooke was a person who coined the term cell. In 1670, Antoine van Leeuwenhoek was the first person to observe living cells in the stagnant pond. Most probably he was looking at a unicellular protozoan or algae, but he was the first person who looked through the lenses of microscope, the living cells that were moving in the water, stationary water. In 1833, Robert Brown discovered nucleus in the plant cells and suggested that it is very important for cellular regeneration. In 1838 and 39, Matthias Jacob Schleiden and Theodor Schwann, they proposed the cell theory, and that all plant tissues are composed of cells, and a cell is the fundamental unit of life. That was essentially the cell theory. In 1845, Brown restated the second part of cell theory, proposing that the cell is the fundamental unit of life. In 1855, Robert Virchow, he did the third part of the cell theory, stating that cells only come from pre-existing cells, indicating the significance of cell division, DNA replication. So at this point of time, all the discoveries were on the surface, but they were serving as prompt to dig deeper, to look at the nucleus, to look at the cells, to look at the organelles. So these 5 to 6 discoveries, maybe more, they were the fundamental fountain of knowledge, especially in the biological science that began to sprinkle. So much so that it came to a point in 1865 where a major observation happened. And this observation was led by none other than Gregor Mendel, who worked on the pea plants. So I'm not going to go into the genetics of Mendel's work or a specific work, but what were the take-home message from Mendel's work? The take-home message from the Mendel's work was that characters could be passed on from one generation to the next. So what model system did Mendel use? Mendel used pea plants, and he looked at various characteristics. And what's amazed him that the characters that are being passed on from one generation to the other generation is passed on in a formula. What I meant by that is that, if you consider a breeding of purple flower with white flowers, the first generation is all purple. And in the second generation, when the breeding takes place, there are 3 purple and 1 white flower. So what happens to the white flower? The white flower is not lost, it is hidden in the F1 generation. So what are these features that is passed on from parents to the next generation? Of course, it is in the context of pea plant, but it has a very deeper meaning. And the reason that Mendel is regarded as the father of genetics is for the main reason that these inheritance of characters that Mendel observed in pea, he could see in the human being also, particularly the sex-linked disease. So the sex-linked diseases in humans, they follow Mendelian pattern, right? So that's why so think about it, that Mendel's work, which was on pea plant, could be extended to study human diseases, particularly sex-linked diseases. So from pea to human being, big jump, big observation, big discovery. So Mendel is regarded as father of genetics. Mendel did all his work around mid-1800s, but his work is very relevant in today's time, and we are in 2020. So what happens that when a scientist makes a discovery, then different scientists, they're working on different model system. So they always want to evaluate, test the hypothesis. So when Mendel work, which was a hypothesis, but since it was well established, it became the law of genetics, Thomas Morgan in 1910 wanted to see whether the eye features, in flies. Morgan mated wild type red female with mutant white-eyed male. And he observed similar pattern as what Mendel observed for them. So Morgan then bred F1 red-eye female to an F1 red-eyed male to produce F2 generation. And when you look at F2 generation, you see if you look at the phenotype in F2 generation, what it is the eye color, it is 3:1, very similar to what we observe in Mendel observed in pea plants. So after Thomas Morgan, there was a very famous scientist. Her name was Barbara McClintock. She was working in Cold Spring Harbor, which is in New York, Long Island, and her work was mainly on plants, particularly on maize. But Barbara McClintock observed something very phenomenal. She observed recombination, right? what is recombination? Recombination is a process by which DNA could exchange part, or chromosome could exchange part. So she investigated this in plants, particularly maize. And I have had a chance to visit her lab in Cold Spring Harbor. Um, her lab was essentially like a home. And in the backyard, she had a field in which she used to grow maize, and she used to spend a lot of time with this plant. And what observations she made, that plants undergo recombination. And what she proposed a very unique thing. She proposed that this recombination could be because of transposon. Transposon we will study in mutations, but transposons are present in this maize plant that can jump from. Transposons are fragment of gene that could jump from one location of the chromosome to another. And this could create new series of plant. Now I want you to extend your thought. In today's time, we are using, we are consuming a lot of genetically modified plants, right? These genetically modified plants have been produced in labs by some biotech companies. But what I want you to emphasize that since plants are in open environment, they have tendency to undergo recombination. It could be cross-pollination, it could be transposons. So plants have natural way of undergoing recombination. So that was the great work of Barbara McClintock, and she got the Nobel Prize in 1983. And she did a phenomenal work. Second scientist that I would like to, I want you to look or admire is Frederick Mischer. You'll be doing BIO461 lab, and you'll be isolating DNA or extracting, nucleic acid. So this breakthrough of extracting nucleic acid from the cells was first done by Frederick Mischer. And he could separate the cell wall, he could separate the cytoplasm, and in the nucleus, he noted there's a pus-like chemical. Now you will notice that when you extract DNA and when you purify DNA, DNA looks very thick, very jelly-like. It may not be pus, but it's very jelly-like. It's very wool-looking, and so he named this substance as nuclein. That became the precursor for nucleic acid. So what did we discuss until now? We discussed name of many scientists and their contribution. We discussed about Mendel, Thomas Morgan, Barbara McClintock, and then Frederick Mischer. It is as a student of biology, whether it is me or you, it is very important to know the ladder in which biology developed. And you think that if biology was not there in today's time, and there would have been this pandemic, how would we know what is going on, right? So fundamental knowledge of any discipline, of any subject is very important. And the starting point is the history. Now, this is very important, and this is the work of Fred Griffith. And Fred Griffith, he did the work on Streptococcus pneumonia. So Streptococcus pneumonia, this is a bacteria that can cause pneumonia. So let's call it as Streptococcus pneumonia. All these scientific names should be italicized, but because I'm using Zoom and this doesn't give me an option to italicize, so I'll just write it this. So this is Streptococcus pneumonia. This can cause pneumonia, and this pneumonia can kill the mice. Now, what happened that Fred Griffith, he noted two strains of this bacteria. One was smooth strain and the other was rough strain. Smooth strain and rough strain. Now, when he did experiments carrying forward to understand where this genetic material is located, what is its function, does it have any significance, and I'm talking in terms of genetics. I'm talking in terms of DNA. So when he took the smooth bacteria, smooth Streptococcus pneumoniae bacteria, and he injected into the mice died, right? So it's a clear case that, okay, the bacterial strain was injected, the mice died, means that it was a disease and mice died. Now, if he took the strain, rough strain, the mice lived. Mice did not die. So same bacteria, but two different strains had dramatic effect. So any scientist who come across this such as dramatic results would like to know what is going on. So what he did, he took the smooth bacteria and using high temperature, he could kill the bacteria, and when he injected into the mice lived. What does it suggest? This experiment suggests that there was some factor in the cells of smooth bacteria that is responsible for causing the disease. Disease pneumonia in the mice. So what he did to understand what is going on, he took heat killed smooth bacteria, and he mixed this bacteria with the rough bacteria and then injected into the mice. And again, he saw that all mice died. All mice were dead. So the billion-dollar question was, what's going on? So essentially what he did first experiment, smooth bacteria, it could kill the mice. The rough bacteria did not kill the mice. When he mixed the heat-killed smooth bacteria and rough bacteria, the mice again got killed. What does it suggest? That something was present in the smooth bacteria that transformed the rough bacteria. Now here I would like to talk about one term. That term is called as virulence. Right? Virulence means the potential to cause disease. So smooth bacteria was virulent, meaning that it had caused disease in mice and the mice could die. The non-virulent is the bacteria or any pathogen or bug that cannot cause disease. So the animal or the host will live and will be healthy. So what I want to connect this to is that the smooth bacteria was virulent strain, and the rough bacteria was non-virulent. But something happened that transformed. There's some factor that would transform the smooth bacteria some factor in the smooth bacteria would transform the rough bacteria. So what was that factor? Right? So this was done by another scientist. His name, this was done again by a group of scientists, and their names were, they worked as a team: Oswald Avery, McCarthy, and McLeod. They were the scientists in New York, in Manhattan, in Rockefeller University, which is located on 68 and New York Avenue, just near where Sloan Kettering Memorial Hospital is there. So what these scientists did, they took the smooth strain, right? They took the smooth strain and they extracted RNA, protein, DNA, lipids, and carbohydrates. So what they did, what these scientists did, they took the smooth bacteria, and then fractionated this bacteria into major biomolecules. For example, RNA, DNA, protein, and lipids, and carbohydrate. He took this smooth bacteria and isolated these factors, and these factors were RNA, DNA, protein, lipids, and he took these factors individually and injected into mice, right? And he injected these into mice. So here he noticed only that when DNA is used as a factor, then this DNA is responsible. DNA is responsible for converting, I should use the word transforming, converting the dead smooth bacteria when put together with rough bacteria to kill the so DNA was that factor. So what I want you to emphasize here is to remember what I mentioned, that it's not possible for one scientist to answer all the questions, but it occurs step by step. One scientist does the best, but his work raises more questions that is taken over by next generation scientists. It could be colleague or it could be student, stuff like that. So what was clear until now? So if you go back and if we revise, so scientists discovered microscope and they could look at cells. They coined cell theory, that every organism is made up of cells. Cells are the building blocks, and it is the cells that give rise to new cells. Then came the discovery of Mendel for looking at characters that can be passed on. This was confirmed by Thomas Morgan, and then plant recombination by Barbara McClintock, and then Fred Griffith, and this particular experiment by Oswald and Avery. So now it was pretty much sure that DNA is the genetic material, but more proofs were needed, and we needed to study this in different model system. So now here, I would go into a little bit of chemistry, and what I would like to introduce you to is that radioactivity. So radioactivity is also known as nuclear delay. Nuclear delay or radioactivity in the process in which a nucleus of an unstable atom loses energy by emitting ionizing radiations. So these atoms are a little bit unstable because they carry an extra particle, and because of that instability, they can release energy, and that energy can be trapped to understand various biological processes, right? And that energy can be captured, and we can come to know, and once you capture the energy, you can quantitate how much energy is gained, lost, and so on and so forth. So let me take some examples. So these atoms, which are high energy atoms, unstable, they have same number of protons, same number of electrons, but they have varying number of neutrons. And such atoms, they are called as isotopes. So for example, if you look at carbon, right? So carbon has atomic number 8, which means number of protons, and it has 8 neutrons. The total is 16, right? So here, if you see oxygen, uh, the neutron is 9. It should be 8. Right? Similarly, in a different oxygen, it could be oxygen 18. So going back, so for example, phosphorus has an atomic mass of 31, but due to presence of an extra neutron, the atomic mass becomes 32. And this phosphorus 32 becomes an element or an atom with high energy. And this energy of phosphorus could be trapped to discover the more detailed structure of DNA. Similarly, you have nitrogen. Um, Nitrogen has an atomic weight of 14, but if there is an extra neutron, then nitrogen becomes N15, right? And this N15 nitrogen it releases extra energy that can be tapped to do various biological experiment. So this is chemistry, isotope, but these chemical atoms or elements can be used to answer biological questions. Now let me tell you why and how. So, so we all know by now that at least superficially, that DNA is a double helical molecule, right? And the DNA has the DNA is a actually a macromolecule, which is called as polynucleotide. Now this is a macromolecule. They are made up of individual repeating units. So what is the monomer? What is a single unit? It is a nucleotide. Now, what is nucleotide made up of? Nucleotide is made up of sugar. What sugar it is? This is deoxyribose. Then it is, and what is deoxyribose? What atoms are present here? It's carbon, hydrogen, and oxygen. We will go into this later on, right? Then the second molecule is phosphate. Right? Phosphate is phosphorus and 4 atoms of oxygen. Phosphate. Then the third is nitrogenous bases, which are essentially purines and pyrimidine. And what are these nitrogen bases made up of? They are made up of carbon, hydrogen, nitrogen, and oxygen, right? So the DNA is the genetic material in our cells. It carries information from one generation to the other generation. DNA is present as chromosomes, right? But what is the fundamental building blocks of DNA? Yes, polynucleotide, nucleotide, but what is the building block? It is carbon, hydrogen, oxygen for sugar. For phosphate, it is phosphorus and oxygen. For nitrogenous bases, which are purine or pyrimidine, are nitrogen, carbon, hydrogen, and oxygen. So if you focus, the point I'm trying to make here is that we can use phosphorus as an isotope, high energy, P32. We can use nitrogen, N15, which is a high energy molecule. So the regular nitrogen is N14, 1,4. But high energy nitrogen will be N15 with an extra neutron. Similarly, we can use carbon 13, right? Carbon has an atomic weight of 12, but if there is an extra neutron, it will be carbon 13. Hydrogen, it could be tritium, right? Oxygen could be heavy. So there are various possibilities by which we could use isotopes to study the role of DNA. Remember, what is the role? To establish that DNA is indeed a genetic material through different approaches, right? So, but what scientists did, what model system they used? They used a model system of bacteria, right? So this is a bacterial cell, and you know that these bacteria could be infected by, these bacteria could be infected by a special virus which is only present in bacteria, and those bacterial infecting viruses are called as bacteriophages, right? So bacteriophages. Now, the question here is that if bacteriophages are infecting the bacteria, then what is the substance for killing the bacteria? What is the substance for propagating the bacteriophages, right? So what scientists did, they used radioisotopes here. What radioisotopes scientists they used? They used P32. P32, why? Because DNA has, as I mentioned to you, phosphate, and phosphate is part of DNA backbone. Second isotope that they used was S35, sulfur 35. This sulfur 35 could bind to an amino acid cysteine or methionine, right? The phosphate group can only bind to. Why? Because DNA has a phosphate backbone. So what scientists did, they took two types of bacteriophages. One labeled with P32, and the second labeled with S35. So first labeled with P32, and then second labeled with S35. So scientists could label both the DNA and the proteins, right? And they took these two kinds of viruses or bacteriophages and put together with the bacteria. What bacterial model they used? They used E. coli, Escherichia coli. Remember, bacteria is a pathogen that can cause disease in human body, but bacteria has served as an excellent laboratory to understand various mechanisms of molecular biology that we will study more. So most of the molecular biology work, it has been done by using bacteria as a tool, particularly E. coli, right? So keep that in mind. So they took the viruses, the bacteriophages, they put the bacteria, made a mixture, right? And bacteriophages, they infect bacteria very fast. And these bacteriophages try to use the resources of bacteria for its own benefit, right? So what scientists they did, they took the bacteria and and made a mixture of bacteria plus infected bacteria with bacteriophages, and this centrifuged. What do I mean by centrifuge? Is that so? What scientists they did? Uh, they they took the bacteria plus bacteriophage in a test tube, and this test tube, this put this test tube for centrifugation. So what is centrifugation? Centrifugation is a force that helps to let the heavier elements to settle down in the tube and the lighter element to float. So let's say if this is a centrifuge, then this is a tube which has which is having bacteria and bacteriophages, and these bacteriophages have been dually labeled with P32 and uh, and uh, and proteins with S35. So what scientists noted after centrifugation, that that all the some particles they settle down in the bottom of the tube, and there were some particles that were floating on the top, right? And then what scientists they did, so this experiment was performed by Hershey and Chase, right? And they used T2 bacteriophages, and they used P32 and S35. So when they separated the supernatant and the pellet, they found that the pellet, which is rich in P32, meaning the DNA, was the reason for infectivity, right? So going back to to this, so scientists what they did, they took the the material that settled down, which was P32, and the material that was floating, that is called as supernatant, and then incubated with bacteria. So scientists, they could find out, they could discover that it's the DNA that is responsible for the killing of bacteria. So in 1950s, some insight into the structure of DNA was established, the physical structure, and this was mainly by Linus Pauling from California, and Maurice Wilkins and Rosalind Franklin, right? So in London, right? So one was physical chemist, and other were X-ray crystallographer, which is a unique technique to look at the structure of DNA. So now I'll go into specific details of the structure of DNA. So we all know, as I mentioned to you before, that DNA is a double helical structure, right? Double helix means it is made up of two helix, double helix, right? Now, what is important here to know that the structure of DNA is double, as I mentioned, double helical, means double-stranded, and most importantly, this is anti-parallel. What do I mean by anti-parallel is that, so let's say for sake of understanding, I have drawn two strands which are through two different colors. One is yellowish color and the other is red color. What is meant by anti-parallel is that one strand, it goes from 5 prime, it goes from 5 prime. So I'm talking here on yellow strand, it goes from 5 prime to the 3 prime. Right? And the other strand, which is red color, it goes from 3 prime to the 5 prime. Right? So if you focus here, two strands, anti-parallel, both are different in terms of their orientation. One is 5 prime here and what is 3 prime here. Similarly, if you look here at the other end of the DNA, 5 prime and 3 prime. They are not both 5 prime and 5 prime or 3 prime and 3 prime. They are 5 and 3. That makes them as anti-parallel. Now, how does this happen, right? So let's look a little bit into nucleic acid, right? So nucleic acids, it is of two types, right? DNA and RNA. Right? So DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. Now, what is the major difference between DNA and RNA? Right? What's the major difference? So one of the major difference is that it lies in sugar. So, let's understand this. I'm trying my best to draw the structure of sugar, but I'll come back to the slide, but try to understand that both the sugar in DNA and RNA, the sugars are ribose. Ribose is made up of 5 carbon sugar. Structure something like this. It's not perfect, right? So this is carbon number 1, right? Then this is carbon number 2. Carbon number 2, then carbon number 3, then carbon number 4, and then there is a carbon number 5 here. It goes something like this. It's not perfect, so I beg your pardon. So the carbon number 5 is somewhere here, right? Now I want you to focus with all your attention on carbon number 2. This carbon number 2 in DNA is only H. And this, interestingly, this carbon number 2 in RNA is OH. So one of the major differences between DNA and RNA lies in the presence and or absence of oxygen on the carbon number 2 of sugar, and that sugar is ribose. And this creates two different worlds, DNA and RNA. So I repeat, DNA is deoxyribonucleic acid with lack of oxygen on carbon number 2, and RNA has oxygen as OH group on sugar ribose. So let's go back to the PowerPoint slide. So what you see here in this structure in this structure is a sugar ribose, 5 carbon atoms, and this sugar, so let's focus on carbon number 5 prime and then 3 prime. So this 3 prime carbon is attached to phosphate group, right? And this phosphate group on one hand is attached to 3rd carbon atom, and on the other hand it is attached to the 5th carbon atom. So let's go back to the drawing board. So the backbone of DNA, this is the backbone, the ladder. This is called as backbone. This backbone of DNA is made up of sugar and phosphate. This backbone is made up of two things. Number one, ribose sugar. The second thing is phosphate. Now the question comes, how are they attached? So they're attached by a covalent bond called as phosphodiester bond, right? So what happens here is that, so let's draw another two sugar moiety of DNA. So let's say this is one sugar, right? And this is the second sugar consecutively. So how these two sugars are joined in the backbone of DNA. So this is connected by a phosphate molecule. So how it is connected? So here, this is carbon number 3rd, and on the second sugar, carbon number 5, right? So this phosphate molecule, on one hand joins to carbon number 3rd, and on the other hand it joins to carbon number 5th, right? So this is referred as phosphodiester bond, right? Why diester? Because it forms two ester bond, one ester bond with 3rd carbon atom and the second ester bond with the 5th carbon atom. Phosphodiester bond, two ester bonds, right? That is formed by phosphate and the two consecutive ribose molecules on DNA. And this is what you see in this figure. So pay attention. So one sugar ribose is connected to phosphate, and this phosphate is connected to the next sugar. Now, where does the nitrogenous bases comes? So the nitrogenous bases, let's go back to the slideshow. This nitrogenous bases, they are connected to carbon number 1 through glycosidic linkages, right? So these nitrogenous bases could be purine and pyrimidine. So purines are purines are adenine and guanine, and the pyrimidines are are thymine and cytosine. Right? Now here again, there is second difference between DNA and RNA. Second difference is that RNA do not contain thymine, instead they contain uracil. Now you can, so the backbone of the DNA is phosphodiester bond. Now the question comes how these two double helices are joined, right? So these two double helices, they are connected, right? They're connected. So how they are connected? They're connected by nitrogenous bases. So what's the rule? The rule is that adenine forms double bond with thymine and guanine forms triple bond, uh, triple bond, triple bond with guanine forms triple bond with cytosine, right? So that's essentially the structure of DNA. DNA is double helical molecule. They're anti-parallel. The DNA has double stranded is made up of sugar and phosphate that are connected by phosphodiester bond. And these two strands are connected by nitrogenous bases. Purine bonds with pyrimidine, for example, thymine bonds with adenine, and guanine, that is pyrimidine, guanine, that is purine, forms bond with cytosine. Now I'm saying the word bond. So what is this bond? This is hydrogen bond. So, so what kind of bonds do you know so far? So there are covalent bonds, there are ionic bond. So covalent bond is because of sharing of electrons. Ionic bond is because of attraction between opposite ions due to loss or gain of electrons. What are hydrogen bonds? Hydrogen bonds are bonding attraction that is based upon the distance, right? So hydrogen bond is the weakest bond compared to ionic and covalent, and it is formed because of proximity of distance between the two strands. So A with T, G with C. Now you can ask me a question, why A with T, why G with C, why not G with G, why not C with C, why not T with T, why a purine always pairs with pyrimidine? That's because it helps the DNA to be stable, right? Because the pairing between purine and pyrimidine, the diameter of DNA is constant throughout. But if it was GG or AATT, then DNA would be very, very unstable. So it's nature that has divide. Remember, DNA is genetic material. It is important. If the DNA gets mutated, then it's a disease, severe diseases. So nature has tried to make DNA best possible stable molecule. So that's why whenever there is investigation, forensic investigation, the forensic scientists, they go for DNA because compared to proteins, carb, RNA, DNA is most stable, that is preserved from one generation to the next generation in various species, if you note, not only human being. So we understood a little bit the chemical structure of DNA. So now let's look a little bit more deeper into phosphodiester bond. So this is what I mentioned, that phosphodiester bond here, if you see, it is connecting to the third carbon atom, right? It's connecting to the third carbon atom. and the second is connecting to the fifth carbon atom here, right? And this carbon atom, the carbon number 1, is attaching to the nitrogenous bases. Now, another very interesting theory was proposed, and this was called as Charagaff rule, which was proposed by Irvin Charagaff, right, in 1947. He mentioned that the percentage of A means, so let me go back to the drawing board. So Irvin Charagaff, he proposed a rule that said that in a DNA of any species, the percentage of adenine is equivalent to the percentage of thymine. And similarly, the percentage of guanine is equivalent to the percentage of cytosine. So now this can be extended into a quantitative problem. So let's say if I give you a question and I say that there is an organism that has been discovered now, it has guanine to be 23%. So the question I will ask you, what is the percentage of adenine, right? So how to calculate? So it's very straightforward. So if guanine is 23%, then cytosine will also be 23%, right? Because what is the Charagaff rule? That percentage of G is equivalent to percent of C. So what's the total? Will be 46. So 46 percentage in the species of a DNA is C plus G. So what's remaining? So let's say what we can say that let's say out of 100%, 46% is G plus C. So what's remaining? The remaining is 54. So we know that the percentage of A is equal to percentage of T. So from here, we can back calculate it that 27% of this will be adenine and 27% of this DNA will be thymine. So if we know the percentage of one of the nucleotide, one of the nitrogenous bases, you can calculate the percentage of all 4 nucleotide. So that was Charagaff rule. Now, let's go into more detail how the the structure of DNA was discovered. Now, scientists around the world, mainly the research that was going in US and Europe, they were confirmed that DNA is a double helical molecule. It is a polynucleotide, and there's phosphodiester bond, there is nitrogenous bases, but scientists were not able to prove how the double-strand DNA are connected, in meaning that what we know today that A is connected with T and G with C in a double helix, scientists did not know that this connection is through hydrogen bond. And remember what I said shared with you is that the hydrogen bond is a function of distance. So there are very many scientists were in race. For example, in US, Watson and Crick pair, and in Europe, very meticulous work, very hardworking, very smart, genius work was being led by Rosalind Franklin and Maurice Wilkin. And what were they? They were X-ray crystallographer. So what is X-ray crystallography? So what this technique does is to, let's say, freeze the biomolecule, capture the biomolecule as crystals. And on this crystal, they throw X-ray diffraction, X-ray light, X-ray beam, a beam of light. And then it creates a shadow, it creates a diffraction. And from there, from that diffraction, scientists can calculate the distance between 2 atoms, atomic distance, so on and so forth. So the work of, I underline this with bold letters, that it's the work of Rosalind Franklin, had a specialty, had expertise in X-ray crystallography, that she established the two strands of DNA which are connected by nitrogen bond. Those are connected, to connect nitrogen bonds. It is hydrogen bond, was proved by Rosalind Franklin. She was the pioneer scientist. And that work was interpreted also by Watson Crick. And so Watson and Crick, they got the idea from the work of Rosalind Franklin, and then they discovered the structure of DNA, and they got the Nobel Prize in 1953. So the big contribution in the Nobel Prize of Watson and Crick was the work of Rosalind Franklin. And what was her contribution? Her contribution that based upon X-ray crystals of DNA, she could propose that the two DNA strands are connected by hydrogen bonds. That's how we see that A with T, hydrogen bond, two hydrogen bonds, G with C, triple hydrogen bond, right? So, so let's focus here. So what I want you to draw attention here is that biology, right? There is a cell, there is a nucleus. Within the nucleus, there's a chromosome, and within the chromosome is DNA. And DNA is made up of nucleotides, sugar, phosphate, and nitrogenous bases. But look at the planning of nature. DNA is very much a physical quantity. It has exact symmetry. It is very stable. It is a structural molecule. It is well engineered molecule. Why I say that? Number one is that the diameter of DNA is constant, right? The diameter of DNA. And also what I want you to focus here is that let's say if this is the DNA, right? So let's, so this is the one of the DNA strands, right? And the second strand is like this, right? So from the distance from here to here, right? This is called as one turn of DNA, right? It's not a perfect picture what I drew, but I'll try to make you understand that DNA is something like this. This is single strand. The second strand would go like this. Right? So this from distance from here to here is one turn, right? And then from here, second turn, right? So the number of nucleotide in one turn of DNA is 10 nucleotides. 10 nucleotides. In one turn of DNA, there are 10 nucleotides. Now, the question comes, what is the distance between two nucleotides? So the distance between two adjacent nucleotides is 0.34 nanometer. So let's go back to the drawing board. So one turn, the distance of one turn is 34 nanometers. And the distance between two adjacent nucleotide is 0.3, or let's say it is 3.4 nanometers. Meaning that in one turn, there are 10 nucleotides. So in one turn, there are 10 nucleotides. The total distance, total length of one turn is 34 nanometer, and the distance between two adjacent nucleotide is 3.4. Right? So here, if you note carefully, this is this is one turn of DNA, 3.4 nanometers, and the distance between two bases is 0.34 nanometer. Right? Now, look at the architecture of DNA molecule. Right? Because of this turns in the DNA, if you look at more carefully, so I'll go back again to explain this to the whiteboard, is that if you look at the way the two strands are intertwined, it creates groove, right? What do I mean here is that, so let's say this is the groove, right? So this groove is deep. This groove is deeper. And the opposite groove, which you can say this one, this is not deep as this one. So this one is not that deep. So let's call this groove as major. The deep groove is called as major groove, and the shallow groove is called as minor groove. Right? As minor groove. So what I mean by major groove and minor groove is that when DNA is, what is DNA? Double helical structure, right? When the two strands intertwined against each other. So let me draw again. So let's say this is the one strand, right? This is the one strand. And let's draw the second strand, right? So something that's not perfect, right? Right? So see here. Pay attention here. So this is a deep cleft, deep groove, right? So because of that deepness, it is called as major groove. Now, if you focus at the opposite side, it's very shallow. It's not that deep, right? It's not that deep. It is very small. So this is called as minor groove. So because of the intertwining of strands in the DNA, it creates two types of groove, a major groove and a minor groove. So this is what is shown in this figure, in this slide. This is what you see here as the red, white, and bluish is balls. They are different atoms. But what I want you to notice here is that what I want you to notice is that so this is So this is a major, this is a small one. So this is minor, and this one is bigger groove. So this is bigger, if you see. This is major, and then again, if you see here, this one is minor, and then again this is major. So the intertwining of DNA against each other, it creates groove, and these grooves are major and minor grooves, and they alternate each other. So one minor, then one major, then minor, then major. Okay, and in this slide there is some details about the major groove and the minor groove that you can study in your textbook. So this is another figure which perhaps might be more clear. So major groove is deep, whereas minor groove is not that deep. And so now the question comes, why there is a major groove and minor groove? It's because DNA serves as a substrate for replication, transcription, and many proteins, they need to access the DNA directly. So these pockets, major groove, they help enzymes like DNA polymerase, RNA polymerase, and other transcription factors to initiate the process of replication or transcription. So it helps in protein-protein interaction. Next slide is very important slide, which says that there are three forms of DNA. B form DNA, which is a native DNA, A form of DNA, and Z form of DNA. These three forms of DNA, they differ, or they could differ in 5 parameters, based on 5 parameters. Helix sense. So B DNA and A DNA, they are right-handed helix, whereas Z is left-handed helix. As I mentioned to you, the native DNA is the B DNA. There are 10 bases per turn, whereas A DNA and Z DNA, they have 11 and 12 bases per turn. And then there's a vertical rise. I would explain later how this vertical rise takes place. So in B DNA, it's 3.4, in A DNA, 2.56, and 3.7 in Z DNA. Helical diameter, right? The helical diameter of B DNA is 90 nanometers, and of A and Z DNA is 23 and 18. So you must watch this YouTube to look at the various biophysical characteristics of DNA. So here represents 2 kinds of bonding or hydrogen bonding, thymine and adenine. They form double hydrogen bond, and guanine and cytosine, they form triple hydrogen bond. In RNA, there is no thymine or uracil, so the bonding is between adenine and uracil in case of RNA. Here also, same thing. As I mentioned to you, DNA is antiparallel strand. Why antiparallel? It is because the orientation of the strands, 5' to 3', and then 3' to, so one strand goes from 5 to 3 strands, 5 to 3, and the other strand goes 3 to 5. So this strand goes from 3 to, so one strand 5 to 3, the other strand 3 to 5. And the phosphodiester bond connects two neighboring 2 adjacent sugar through bonding with 3rd carbon atom and the 5th carbon atom. Carbon number 1 forms bonding with the nitrogenous bases, and 2 nitrogenous bases, they form hydrogen bond. And who discovered these hydrogen bonds? was Rosalind Franklin. What was the technique she used? X-ray crystallography. Right? Now, so by now, it was established, starting from the discovery of microscope, cells, cell theory, nucleus, Mendel, Morgan, Barbara McClintock, Fred Griffith, Oswald and Avery, Hershey Chase established that DNA is the genetic material. So now it was established that now the question, these experiments, these data led to more questions. And the second series of questions were, if DNA is important for a cell, being its genetic material, how does it replicate? But the question of replication comes why? Because cells divide, right? So it was established in cell theory by Schleden, Schwann, and Virtue that cells could divide, but cells, cannot divide unless it gets instruction. So there must be some programming within the cells. There must be some material within the cells that might be extremely critical. And we all know that it was DNA. The question comes at that time, what scientists were thinking about it and how they proved it, or how they disproved that protein is not a genetic material, lipid is not a genetic material, or carbohydrate is not. Why all the attention focused on one molecule, that is DNA. So now the very elegant experiment was performed by Messelson and Stahl, right? What experiment did they propose? They proposed that the replication of DNA is semi-conservative. And let me explain to you what semi-conservative means. So DNA, I'm drawing, not drawing the helix, I'm just making it very simple. So DNA, as I mentioned to you, is double helical structure, right? It's made up of two strands, right? For sake of understanding, we make these two strands as colored, two strands, right? And they are anti-parallel. What do I mean by anti-parallel? Is that one is 5' and 3'. So these two strands are 5 and 3'. And these are 3 and 5'. So they are double helix anti-parallel, connected by nitrogen spaces. Now cells have to grow, but they can't grow unless the DNA divides, because all the information is lying in the 4 bases of DNA. So how does DNA replicates? So, and more important is, how did the scientist discovered that it is semi-conservative means of replication? So if this is the body of DNA, then the question is, how do we know the mechanism of replication? So there are 3 possibilities, right? There are 3 possibilities. There are 3 possibilities. One, that DNA replicates in a conservative way, right? So DNA replicates in a conservative fashion. The second option is that it could be replicating in a semi-conservative. And the third is dispersive model. Nothing sure, something like mix of conservative and non-conservative, There is no structure, there is no organization, right? So these are the three possibilities in which DNA could replicate. So another question comes, what are the tools that scientists used to answer this question, whether the DNA replicates via conservative way, semi-conservative ways, or dispersive way? So here, scientists, they went back to a radioisotope called as N15. Right? So N14 is the native form of nitrogen, and N15 is in the isotopic form, meaning that they have an extra neutron that can release energy in the form of gamma radiation that can be captured, and it can prove an experiment, right? So in other words, I can say that in our cells, we have DNA in the nucleus. The nitrogen that is present in our DNA is N14, right? But if we take out our cells and grow those cells in a lab condition, and we put nitrogen 15, which is a radioisotope, some of the DNA, some percentage of DNA will become N15, because DNA or enzyme cannot distinguish between N14 versus N15. So scientists used N15 to understand the mode of replication of DNA. So let's go to the slide. What did scientists do? So scientists, they grew. So let's start from the top. So scientists, they took, so bacteria, they like nutrition, right? And nutrition, you will see the bacteria they like most is LB medium, or a scientifically synthesized medium, nutrition medium that bacteria can grow. So scientists, they grew this bacteria in N15 medium, means this nutrition, they had nitrogen 15 and not N14, which is native, right? Then after growing this bacteria in N15, scientists then switched the bacteria to N14, meaning that they grow the bacteria in N15 in radioisotopic condition, and then they switch the bacteria to N14, which is a native nitrogen. And then they collected these bacteria and try to quantitate whether the division of or replication of DNA is conservative, semi-conservative, or dispersive replication. So what is meant by semi-conservative, right? So semi-conservative is this form of replication, right? So this is the parent DNA that is grown on N15, and in first generation, the DNA will be hybrid of N4. So in first generation, or let's say call it as generation 0, because the cells are growing in N15, all the bacterial DNA will be N15. And in generation 1, when it is switched to N15, the 50% of the DNA will contain N14, and 50% will be N15. So this will be hybrid. And then when you grow further in second generation, you will get 4 copies, 50% semi-conservative and 50% 100% N14. In the conservative model, what will happen is that in the semi-conservative model, 100% will be N15 in generation 0 because obviously they're growing in N15. Then in generation 1, 100% of the DNA will be either N15 or N14, right? And in the second generation, 75% of the DNA will be N15 and only 25% would be N15. And dispersive model is something like it's mix and match, right? So how did the scientists, they figure out? And remember, I mentioned to you that N15, which is radioactive, can be quantitative. It could be using various physical instruments. It could be calculated. So scientists, long story short, scientists using N15 as radioactive isotope, they could figure out that the mode of replication is semi-conservative. And at the bottom of this slide, there's a YouTube link that you must watch. Right? So if we go back and trace about what we learned until now, first, from cells to nucleus to chromosomes to DNA to the mode of replication, which is the semi-conservative mode of replication of DNA. Right? Another thing that is very important that I would like to share with you, that we will learn later, is that the genetic material in an organism could be DNA or RNA. Right? DNA, let's say in most of the mammalian cells or animal kingdom, plant kingdom, prokaryotes, the DNA is the genetic element. RNA could be genetic elements in viruses such as HIV or COVID-19, right? They have genetic material as RNA. So genetic material could vary, right? But what is constant, whether it is viruses or bacteriophages or bacteria, is the genetic code. Right? Genetic code is a table of 64 triple nucleotides, right? Triple nucleotides that correspond to a single amino acid. Right? Single amino acid. Each triplet nucleotide corresponds to a single amino acid. Right? So why I make this point here is what is the central dogma of biology? The central dogma of biology is that DNA is the genetic material that serves as a template for RNA, and RNA serves as a template for proteins. Proteins, right? So DNA to RNA, this process is called as transcription, and RNA to protein is called as translation. Now, what I want to highlight here, that 4 bases of DNA, ATGC, can give rise to 4 bases of RNA, AUGC. But what is most interesting is that these give rise to proteins which are made up of 20 amino acids. So the ratio doesn't fit. How could 4 bases of DNA that transcribes into RNA, and RNA 4 bases translates into protein, the ratio 4 to 20 doesn't match up. So who was the scientist who made this understand for us? He was Hargobind Khurana. So he said that 4 nucleotides cannot code for 20 amino acids. Even if you have 4 times 4, which is equivalent to 16, this also cannot code for 20 amino acid. So based upon his experiments, he could prove that 4 times 4 times 4 equivalent to 64. So he said that, he proposed that permutation and combination of 3 nucleotides, it forms triple nucleotide on mRNA, and that serves as a genetic code. And he proposed the table. So what was the table? Table was still called as genetic code. So here there are triple nucleotides. Each triple nucleotide codes for a single amino acid. But there's a very important message here. So out of the 64 triplet condition, only one triplet nucleotide, AUG, codes for methionine, right, as a start codon, but there are 3 stop codons, right? So let's go back. What do I mean? So I mentioned to you that DNA can transcribe RNA, and RNA can translate into proteins. So let's say this is the DNA molecule, double helix, right? This DNA molecule gives rise to an mRNA molecule, right? Now in this mRNA molecule, in this mRNA molecule, right? 3 nucleotides codes for one amino acid, and there are 64 triple nucleotides. Why? 4 times 4 times 4, right? 64 situation. Out of this 64 situation, one is start codon, right? Start codon is AUG, that codes for amino acid methionine, and second is that stop codon, right? Stop codon will restrict the size of the protein. There are 3: UGA, UAG, and third is UAA. Third is UAA, triple nucleotides, right? So the starting will be the first nucleotide will be triple nucleotide will be AUG, and the length of the protein will be determined by stop codons UAA, UAG, or UGA. So if I ask you a question that there is an amino acid that is made up of 900, there is a protein that is made up of 900 amino acids. How many nucleotides will code? How many the mRNA will be composing of how many nucleotides that would give rise to 900 amino acid protein? The answer will be 2700, because the ratio is 1 to 3. 3 nucleotides code for 1 amino acid. So we covered a lot, beginning from starting from how cells were discovered, who discovered cells, and how DNA was discovered as a genetic material, semi-conservative mode of replication, genetic code, and a lot of biophysical properties of DNA. So this is what is chapter 1 about the history of DNA. Okay, so I finished this chapter and we are done with chapter number 1.